Welcome back, everybody. We're going to continue looking at the human genome. And the book starts off here noting very correctly that something like 99%, or give or take, of all DNA based sequences are identical between all humans. This is because at some point in humanity's history, there was a genetic bottleneck that we went through, and we are all descended from pretty closely related ancestors way back in the day, some 200,000 oddish years, depending on which study you read. But all of us are pretty closely related, genetically speaking. 99% is a high similarity. But some of those differences cause diseases. Some of them cause a loss of function, like low production of melanin, which is why you have people with fair skin, or hypermelanization, people who have too much melanin. Minor alterations, mutations, can cause some real differences or real problems. These variations in the DNA sequences that occur when a single nucleotide in the genome is changed are called single nucleotide polymorphisms, SNPs, or sometimes, depending on which book you're reading, point mutations, single point mutations. But your single nucleotide polymorphisms are responsible for some of the big variations within humans. There's not a lot that has to change because just like a computer operating system, if you swap a 1 for a 0, or you swap a A for a C in DNA, you can change something pretty radically or break it completely. So the HapMap project was developed on the back of the Human Genome Project to find genes that are linked to different ethnic groups around the world. And so like that 23andMe or the Ancestry.com DNA tests use these HapMaps to try to tell you about where your ancestry is and where most of your DNA comes from geographically. So it relates your haplotype or your genetic gene codes to a map of the globe so that you can see kind of where your ancestors came from. And it's pretty neat. But more importantly, it can help you look into those people groups that have hereditary diseases that might cause problems. So these regions of linked variations are mapped out, and you can test or compare other DNA against it. Now, assembling the HapMap project involves the identification of groups of these single nucleotide polymorphisms in a specific region of DNA and from a specific people group, right? So you have to have that single point mutation and the people group that it's most commonly associated with to be able to identify it. So this information is then used to help researchers take the next step to find genes that cause diseases. So more than just figuring out heredity, which is what the HapMap is used for commercially, just for fun, you can help find diseases. So again, I have a disease called Stargardt's disease that has left me legally blind. And so they can use these HNPs and their locations on specific genes in my DNA to then look for it in other people who might be carriers of the disorder. Now, a daydream, a future daydream of science, I don't think this has been really realized yet, is called pharmacogenetics or pharmacogenomics. And this is where you take the study of the human genome or how the DNA works in a human, and you can start doing things like gene therapies, or you can start doing things like custom medicine for different people, because it turns out, and this is pretty true, I think, universally agreed upon, that different people will metabolize, the drugs will affect people differently. Not everyone is affected by the same drug the same way. This is why when you buy a box of any medicine, it has this really long list of side effects. Some of them are actually what they're supposed to cure, which is hilarious. But the point I'm trying to make is that not everyone gets those side effects. In fact, most people don't get those side effects at all. They only get them because of these types of variations in how the body reacts to medicines. And pharmacogenomics could potentially help figure out who is going to be more likely to not do well with a certain medicine or do better with a different one. And eventually, you might go to the doctor, and instead of them prescribing you a medicine that they would prescribe for anyone else, they would do a DNA test, and then they would know exactly how a medicine is going to interact with your body because they've studied its effects on people with your specific genetic markers, and they could prescribe a custom medicine just for you in the right amounts. Because maybe your body is going to use it a lot quicker, and so you need more, or perhaps it'll affect you more strongly, so you need less, and that could be influenced by your genetic markers or your genetics, and they could identify that with genetic markers in a process called pharmacogenetics. All right. So again, gene therapy is another version of this, and this is where they identify a gene that is broken in your body, okay? And then they attempt to fix it. How do they do that? There's a couple of different ways. In adults, they use a virus to insert new DNA into your cells. Okay, it's called a viral vector when they do this. And it turns out that some viruses naturally, like the virus that's responsible for AIDS, the HIV virus, has a enzyme called reverse transcriptase, where it will insert programming of itself into your cell. Well, by editing that, using that enzyme, reverse transcriptase, hollowing out the viral DNA and putting in the DNA that you want, you can then insert the DNA using this enzyme with a virus to rewrite the DNA in a cell. Okay, and that's gene therapy. When you take foreign DNA and you put it into somebody's cells. Now, another way to do it that's pretty new and that's just now being discovered is called CRISPR. And you can directly manipulate DNA with this, and you can do it and then do it to, say, a newly fertilized embryo, a baby that hasn't been implanted into the mother's uterus, and create a child that doesn't have a disease. You can edit out a disease for a child. And that's probably where we're going to be going in the near future. It will be trans or polygenic humans. They've already passed laws in some countries to allow for the use of 3 parents in the creation of a child. There is a specific disease that causes mitochondria to not produce energy properly. And so what has been approved in Britain, if I recall correctly, less than a few years ago, is the use of one mother's egg, having its nucleus removed, meaning all of that person's genetic information is gone except for the mitochondrial DNA, and then having the mother who has the disease have her DNA put into the egg, and then fertilizing it with the husband or the male's sperm to produce a child that has genetics or genes or genetic information from 3 people. It has mitochondrial DNA from one mother, the female gamete DNA from the second mother, or the primary mother, you might say, and then the DNA from the father to undergo sexual reproduction. And they do this to cure diseases. Now, can you use CRISPR to edit fertilized embryos? There are rumors, perhaps unsubstantiated, that I've heard that in some countries like China, they have already started doing this. They've edited, it was a set of twins, is what I read, to be immune to AIDS or to the HIV virus. And I think this is probably going to become somewhat inevitable in the near future because arguing against curing genetic diseases is almost impossible from an ethical standpoint. It doesn't matter how many dangers go along with manipulating human DNA. If you can pretty accurately or pretty faithfully get rid of a hereditary disease by editing DNA, um, it's probably going to happen at some point in the near future. Either your children or your children's children will have the option of having their genome, of having their unfertilized or their fertilized eggs checked for disease, then edited to make sure that they don't have disease, and then implanted via in vitro fertilization, where they take the egg and then insert it into the mother so that it grows into a child. I think this is pretty well going to be a thing, whether it's a good idea or not in terms of the bigger picture, because it's just really hard to argue against. Hey, we have a cure for a hereditary illness. We should probably stop that illness, right? It's already kind of being done in a sort of kind of way now, where a child is tested, and if the child has some severe disease, it can be killed. Well, imagine if you could save the child. Instead of killing it, you just edit out the disease and it comes out a totally healthy kid. Wouldn't that be better? These kind of arguments and lines of thinking are things that you will need to deal with as you grow older, because it's your generation that's definitely going to have to run into it, or I predict is going to have to run into it. All right, moving on, because I pontificated, I talked too much about that. Proteomics. Proteomics is very similar to genomics, only it deals with proteins. And proteins are fascinating because they can become enzymes, which catalyze specific chemical reactions. So just imagine what you could do if you could make a custom biological catalyst that would decompose water into hydrogen and oxygen. You'd have limitless energy because hydrogen and oxygen burn to make water, and very vigorously too. So you wouldn't have to burn fossil fuels. You could just use this enzyme. You put it in a cup of water or in a big tank of water, and it comes out hydrogen and oxygen, and then you can cool that and compress it and then burn it later for fuel. That would be phenomenal. It'd be absolutely amazing. And that's just the tip of the iceberg. The number of things you could do if you could create custom proteins, because you understand them and how they fold and how they interact and how they do what they do, well then the possibilities are pretty limitless. It's a really big area of research. And so proteomics is just like genomics. It uses computers and comparative analytics to figure out the differences between protein structures, how they fold, and their functions, and ultimately is going to try to lead to custom proteins. I don't think I need to say any more about that, so I'll keep going. All right, so back to our question, and I'm going to close here because I think I said everything I want to say. Why does the Human Genome Project continue to be significant? Well, it continues to be significant because it turns out decoding the genome was just the start of the problem. Just having the program doesn't mean we understand it and how it works, because the human genome, it turns out, is a lot more sophisticated than we first thought. Genes, the one gene, one protein paradigm has gone away, and it looks like things like gene regulatory networks, RNA-zymes that cause post-transcription editing to produce multiple variants of a protein from a single gene. They cause a much more elegant and sophisticated pattern of development than we first thought. And so it's going to take a lot more research and a lot more time to really understand human genetics, human disease, and how we can use this knowledge for the benefit of mankind in a way that doesn't ruin everything by, causing some kind of catastrophic disaster, some dystopian future in the making. All right, so I'm done. I hope that was interesting. I hope you learned something. If you have questions, let me know. Of course, I love all of the questions. And otherwise, I have some questions for you.